Assalamualaikum. How are you today? We hope you are doing good. Today my group will present for Find a Gene project. Oh, by the way, my name is Atika, and with me Izzat and Kaide. After having a brainstorming, our group decided to discover more about Tourette Syndrome. Tourette Syndrome is the tics disorder which describes muscle groups that cause involuntary movement, sudden, repetitive, and vocalization disorder. We found that the cause of Tourette isn't yet known, but experts do know that genes play part. It's thought that environmental and developmental factors do too. Within the brain, the abnormalities occur in the frontal lobes, cortex, and basal ganglia, as well as the neurotransmitter that help relay messages between nerve cells. We do search in blast about Tourette syndrome and find that the name of protein involved is SLEET and NTRK-like protein 1 found for Homo sapiens species with accession number Q96PX8. Here, we would like to share with you a video that might strengthen your understanding. So, let's watch this video.
Tourette syndrome is a medical condition that people live with in their daily lives. They make involuntary sounds known as tics. It could be a sound or a movement that the body cannot control.
I have this one. Sometimes I click my tongue in the back of my throat like this, but not very often, and I constantly crack my knuckles, even if I already did.
So they mostly would ask me why my eyes keep twitching, or asking me if if I can stop moving my leg.
The tics were more like the head shaking as well, barking. I had a tic for a while where I'd step backwards and walk backwards for a couple of steps and then walk back. I had a tic once when I'd be standing and I'd lift up my leg.
When I was first diagnosed, I was out of school for weeks. My tics were very high. My vocal tics were elevated. My motor tics would really hurt me. It was tough.
7 or so years old, I was responding, well, is this just gonna keep on?
When I was younger, they singled me out and treated me more as as basically almost as if I was an alien in a sense. Because at that point in time, I didn't really understand much about what was happening.
Yeah, I have a 7-year-old. When I take him to school and I go down the elevator, right before we get on the elevator, I'll take my teeth and I'll bang my teeth against the wall. And he asked me, "What are you doing, Dad?" So I said, "Oh, I have Tourette syndrome. It's a condition that causes me to do things I don't want to do." And so the funny thing is, the other day he comes up to me, we're standing there and I don't do it on the elevator. And he says, "Daddy, you didn't do your Tourette's." And I'm like, "No, it's not like that. It doesn't work that way." I have to morph my words. Instead of saying bomb, 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 I go ba or abba.
If I try to suppress them, it doesn't work. That's like trying to hold in a
Young girls would laugh. Kind of embarrassing for an adult to be bullied by young teenagers.
As a young kid, I would tic whatever it was and immediately look around to see who noticed, as if it really mattered.
And then you really start to believe that you are different.
I would be out on the subway and attracting attention from other people, adults, and learning about how cruel people can be. Seeing those kids in DC at the conference, treating their tics as something that was just part of who they are was so inspiring.
It's really cool. Kids don't get judged there, and they have fun with other kids like them.
Other than our tics, we're normal people, because everyone has their own things that make them different from one another. And Tourette syndrome just happens to be one of those things for some people.
Don't be afraid of people with disabilities. Just because someone has a disability doesn't mean that you have to avoid them.
I think the Tourette Association of America means support and community.
My hope is that everyone who sees this video goes home and tells 3 people about Tourette syndrome.
In task 2, we did a TBLASTN search by using the reference sequence of Tourette Syndrome, and we're using BLAST and CBI. And here, as we can see, the results of TBLASTN process, which is this page shown 100 results, which is the highest score is 1376 with the e-value of 0. And we selected the best alignment, which is the highest score, 96.55% of identities and 0 e-value to be our novel query. So in task 3, we did a search and we found some details and information about the Tourette syndrome, the protein that we choose. And here we use the protein ID to conduct BLAST TP in task 4. The protein ID is NP00126843.1. And in task 4, for this step, I conduct the BLAST P to the protein that we got from TBLASTN process in task 3 to prove our gene is novel. For this part, the ref set of NP00126843.2.1 is not novel, because if there is a match of with 100% amino acid identity to a protein in the database from the same species, then this new gene sequence is not novel. A new RefSec is XP00383206.2.1 is a novel gene, because if there is no match with 100% identity, then it is likely that your protein is novel. So in task 5, we did a multiple sequence alignment, where this is the steps of working on MAAFT. The first step is choosing top 10 sequence from the result of BLAST TP. Here we choose the top 10 sequences and then download it and save as seqdump.txt. And then the step 3 is paste seqdump.txt on MAAFT search. Make sure the format is set to ClustalW, as the question requires us to use the format of ClustalW. And on the right side, we can see the pictures of the image of the process. And this is the phylogenetic tree, this is the guide tree, and this is the phylogram.
So, after we do the task 5, which is multiple sequence alignment, we proceed to task 6, which is construct phylogenetic tree using Mega software. So, for the step 1, after align the sequence in MAFFT, download the FASTA file. Or you can use the previous in task 5, which is the seqdump.txt. You just copy paste, you just use that sequence, and then you just change the name and also the and change it, convert it into a .fasta file. And after that, you can align the FASTA file, open the file session in Mega software, and you align into a master. Okay, convert the TXT file into FASTA and align the sequence using Mega software using master. Okay, that's for the steps 2. And the third step is export the result of sequence alignment into Mac file. So, after you export the result of the sequence alignment, you must make sure that you export in Mac file. And then in the fourth step, you just click on the phylogeny and choose the neighbor joining method. And then when you set up for the neighbor joining method, don't forget to choose bootstrap method to get the value for each clade in the tree. So as we can see here, so if you don't do the bootstrap, so you will not get this value, because the function of bootstrap is to get this value. And here is the extra information. I just want to show you the pairwise distance to get the clade value. So this is the pairwise value for the sequence. And then after we done the phylogenetic tree, we go to the discussion for the task 7. So task 7 is the discussion for or summary of what we do in this project. So after we do all the tasks that we been asked for this project, so we discovered that the novel protein that we found, we did a comparison between the original sequence that we obtained from the Tourette Syndrome patient DNA and the novel sequence in the task for SP003832062.1, which is the novel sequence. And then we do the pairwise, which is the chapter 3, the pairwise alignment using BLASTP. And then there are a lot of interesting things that we found. One is this is figure show detail of pygmy chimpanzee. Surprisingly, after we do a pairwise alignment of this two sequence, there are 99.98% similarity between two sequences. From here, our curiosity rises: is human Tourette syndrome brain structure are similarly to pygmy chimpanzees? Therefore, we try to make research about it. We do some search in NCBI also in PubMed. So other than PubMed, we do search in Google. So we found out that a book called Will Save Great Apes. Yes, the name of the book is the Will Save Great Apes, and there are certain related information about the relationship between Tourette syndrome and chimpanzees. This is the screenshot that I get from the book. Thus, chimps will bark involuntarily as they succumb to the tics and spasms of Tourette syndrome of Parkinsonian chimps whose arms and legs undulatedly weirdly from the effects of L-DOPA, and or brain damaged chimps whose gesticulatory cells were imprisoned within the tape loop of acute amnesia. Other than book, but if we look on how we compare the two sequence after multiple sequence alignment and the phylogenetic tree that we construct in the task 6, we found that the both sequence are closely related. How I do conclude that both the sequence are closely related? As you can see, this is the protein that we found in task 3. This is the protein that we found in task 3, and we compare it to this one, the novel sequence in task 4. So as we can see, they are just next to each other here, and the clade value is only 18. So if we divide by 2, which is 18, 9. So somehow it's closely related. And also we do some search in the PubMed, and we PubMed, Gene Bank, and we found some lab related to Tourette syndrome, which is this one. For example, there are one condition, one gene. There are one lab method using deletion and duplication analysis, sequencing, or anti-coding region, and all about Tourette syndrome are using the SLITRK1. That name for the Tourette syndrome sequence. So the conclusion we found out that Lata syndrome had no related at all to Tourette syndrome, but it's surprisingly more related to chimpanzee. Also, we learned that by using TBLASTN, BLASTN, and BLASTP, also various of tools with the right concept, will help us on doing a lot of research. A lot of information that we can obtain from this project, like similarity between Tourette syndrome brain structure and the chimpanzee, are highly similar. We hope that we can learn more about this in future on how to obtain more data to prove of our hypothesis. So thank you and have a nice day.